Hi, I'm Kathi Wagner, and I'm the Executive Director of the Choroideremia Research Foundation. I'm sorry I can't be there with you today, but I hope that I can provide a little background about myself and a little bit of information about the landscape of the current choroideremia research arena. And please note, I am not a medical professional. My background is actually in nonprofit management and fundraising. I've been in various organizations for over 30 years and also in the rare disease network area, trying to extend relationships with pharmaceutical companies and also trying to raise funds to support research for other organizations such as the American Brain Tumor Association and the Cholangiocarcinoma Foundation, which is a rare type of liver cancer. So this is actually something different for me. I've been here about 4 years at CRF, and it's been an education on my part since I don't have anyone in my family actually affected by CHM or any visual disorder, really. So it's been very interesting. And again, this is my perspective as a layperson as to what's going on in the CHM landscape right now. And I think it's fascinating. I think there's a lot of reason for hope and optimism right now. There's an incredible amount of research in various spaces going on. So I'm going to launch a PowerPoint here, and we'll talk about that a little bit right now. So the first, I wanted to mention a recent research paper that talked about new findings. And everybody, I'm pretty sure, is aware now that the choroideremia disease is caused by a defect in the CHM gene. Which initially, many years ago, that was considered to be an easy target because it was a single gene mutation. There weren't multiple genes that were had variants or mutations. However, since that time, they have now learned that there are actually over 250 different mutations that can cause choroideremia on the gene. So it's become more complex in the way that researchers are looking at it. Although there are still some researchers looking to just fix the broken CHM gene, some researchers are now digging in deeper and looking to correct a portion or different mutations. So it'd be a subset of our CHM population so that they could correct, for example, what's called a nonsense mutation, which is in about a third of patients. So there is an opportunity to have various therapeutics that some may affect patients with different types of mutations. So depending also where the defect is on the CHM gene, the outcomes may be better or worse. So they've learned that there's, a alphabetic code basically that causes the CHM gene. And if there's a defect earlier in the alphabet or multiple defects, multiple letters are affected, that this could cause less Rep1 protein to be produced. So this is important to know now. Back again, many years ago, they did not provide all this detail in genetic testing. So it's important to know what mutation you have and where they are on the gene. So if you haven't had genetic testing in about 5 years, it would be good to get a new test because some of the new therapeutics that are coming along could have potential to benefit you or your family members. And there may also be an additional genetic or other factors at play, which could either accelerate or progression or compensate for loss of function. And this is something that we still don't really understand. For example, why some members of a family member progress worse than others. We're doing a study right now on two brothers that both have the same mutation, but one is progressing faster than the other, and we don't know why. Similarly, there are many female carriers who are affected more profoundly than other female carriers, and again, we don't know why. So there might be something else going on, a combination of factors that we could either treat the other thing that is wrong to help add or boost production, or treat that other thing to reduce the impact of the defect. So these are all things that we're trying to understand now. There's so much information, and part of the way that we're doing that is by a group called the International Choroideremia Research Network, or ICRN for short. And this is a group that we've assembled in the last 2 to 3 years. We've now recruited about 140 members from 25 countries. It's a very multidisciplinary group. We have ophthalmologists, engineers, data scientists, biologists, genetic counselors, neuroscientists, and really anyone who has an interest in CHM gene therapy, stem cell therapy, all along the way. We're all getting these great minds together to network and collaborate on various research projects. When companies come to us and they have questions, we're able to assemble some of these researchers together to help them answer those questions, to decide if CHM could be a good target or therapeutic for their product. I've been reaching out to a number of industry companies that are in, near, or around our space, for example, if they're working in gene therapy for retinitis pigmentosa, or if they're doing something in optogenetics that might not currently be in clinical trial, or they're researching for CHM, but we think it might have potential for CHM because it's a disease agnostic type of therapy. Then I'm trying to get our organization and our patients, in front of those companies and researchers so that they might consider adding choroideremia to their pipeline. So we're just trying to stimulate more conversations, stimulate new ideas, and incorporate our patient perspective into the conversations. So right now, the ICRN is focused on several main areas. One is better understanding female carriers. Again, historically, it was just thought that they would have the disease and pass it on to family members. But now we're learning actually many do have symptoms, some of them quite significant, some of them are fully affected. And we want to make sure that those patients are also included in future clinical trials, that they might be eligible to get therapies, and try and understand again why some patients are affected worse than others. And then international data collection. As you can imagine, there's many different institutions around the world, as well as companies that have been studying CHM, but they are keeping their data in silos, right? And they all have their own information, but we're trying to get that via collaboration with other organizations, trying to get them to share that information, de-identified, so it's depersonalized, but just so we understand the mutations, the trends, what mutations or variants might cause worse outcomes or better outcomes, and that type of information. We're also trying to understand the root causes of CHM, which is known as pathophysiology. And we're trying to develop additional animal and cell models. This includes mice and pigs. Right now, these are the primary animals that are used in the space. There is not currently a good mouse model. The one we have was established by CRF funding over 20 years ago, but there's some complications which make it less than ideal for use. So we're trying to develop a better model and also develop a new porcine model. There's not a CHM pig model right now, so it's also called porcine model. And we're trying to use those to help with administration of gene therapy. The size of the eye is closer to humans, and so it's easier to administer gene therapies and injections and so forth, and see how the animals, how the surgical procedures could be improved before going to humans. It's very important that we do this work. And of course, we are trying to find alternatives to animal use, but in the meantime, this is the industry standard. Clinical trial endpoints, that's another thing that we're studying right now, inclusion criteria for patients, who would be eligible, at what ages is the therapy best administered, and what is the best measurement, because as you know, CHM takes so long to progress. Oftentimes it's difficult to measure improvements in a short term. So they're trying to understand if they can use better imaging and other diagnostics, maze tests to determine if a patient is stable or has improvement in the shorter term, rather than observing patients for 5 years or more. And then therapeutics, trying to look at what types of different therapeutics there are. And I'll go into this in a bit more detail now, because again, with 280 mutations and patients in various stages of progression, from young patients who have minimal vision loss to very progressed patients who are blind or only have light perception. It's a spectrum that we're trying to address. And so this is something that, we're not looking just at one patient, we're looking at all CHM patients, right, to provide therapeutics wherever patients may be on the spectrum. So the first that I wanted to discuss is gene therapy. And this is for ideally early stage patients. The earlier, the better, it sounds like, is what we're learning. And these can be administered either behind the eye subretinally or intravitreal, which is in front of the eye. And the 3 most recent clinical trials, all of which CRF supported early research in, have all been in this area. So this included the Nightstar, which then was bought by Biogen. Unfortunately, that failed in a phase 3 in 2021. The Spark trial concluded in 2022. Those were both subretinal delivery, and the 4D molecular therapeutics, which is still underway, is an intravitreal delivery. They currently have dosed all their patients, and they were in a phase 1 trial. They are now awaiting results. So they're observing those patients again for a period of time to see if there are any changes and ensure that it is safe administration as well. That's usually the first goal of a phase 1 trial, is to ensure that it is safe to be administered to patients. The other therapeutics for early to mid-stage patients might include gene editing. And again, right now, there's not anything in a trial for CHM, but this would be DNA editing or CRISPR, which could be making permanent changes, and RNA therapies would be temporary changes to the structure of the eye. And for DNA and CRISPR, it's particularly interesting because they can administer it just to the retina, just to the eye, so it's a very localized, whereas with some diseases, this type of therapy would need to be administered to the whole body. And because it's a small section of the body in the eye, it's more attractive to a lot of researchers to focus on ophthalmic delivery for these types of therapies. And then RNA therapies are, for example, changing, again, the letters. There sometimes might be some switching. For example, in your genetic code, the letter A might be a G instead of where it's supposed to be, so they can switch the letters in theory. And so that is something that is also being studied right now. It's very early. We also have nonsense mutations or stop codon read-through. As I mentioned earlier, about 33% of CHM patients have this type of variant. And so they're looking at how they might be able to administer either drugs like something that would be taken orally, or there might be something that could be a tRNA editing of just that type of mutation. And so then there's some research going on, but it's still very early days for those. And then there's also some adjunctive or eye health therapies looking at cell metabolism, mitochondrial health, oxidative stress, many other types of therapies that would slow potentially progression down. These would not be cures, but could delay progression. And, if it was taken over time or decades, that, it could definitely delay the onset of severe disease. So these are all things that are being studied. And again, not specifically for CHM, but these could have potential for many different eye diseases. And so we're still trying to understand if they might be appropriate for choroideremia. And then we have late stage patients, and these types of therapies include stem cell therapy, which would be sight regeneration. And there are actually many researchers are working on this now, again, because it has potential to treat many different types of vision loss, not just CHM. And so there's an incredible amount of money and resources and time because it would affect so many different people in different disease states. But basically, they are developing eyes in a petri dish. You develop the different layers, photoreceptors, RPEs, like a sandwich in an assembly line. And then they are able to transplant that. And right now they're doing animal studies. We transplant that and see if it integrates and then grows and can restore vision. So again, it wouldn't resolve the original cause of the vision loss, but potentially they could apply one or more of these patches to the eye to help restore a portion of vision for where the patch is applied. The other area they're studying is optogenetics, and this is the delivery of light sensing proteins to the eye. So again, this would restore normal vision. However, it could have a significant impact on people with late stage disease who have very low or no light sensing ability to be able to restore a low level of vision again. So this could be helpful in, for example, navigating and telling the difference if a light's on in a room or if it's night or day or which direction to walk towards a door where there's light. It would be, again, a low level of vision restoration, but it would be a significant improvement in quality of life for people who have very advanced disease. And there's several companies, again, working in this right now in other disease states, such as retinitis pigmentosa, and we are hoping to encourage them to begin including CHM patients in those clinical trials moving forward. There's also prosthetics and robotic vision, which would be artificial eyes. And again, technology's become really advanced, amazing. There's several companies that are working in this space to develop devices that can either be implanted or used in conjunction with gene therapy or in conjunction with a pair of glasses and transmit this information that the artificial eye is seeing to the brain. And so this is interesting because it involves a lot of neurology and how does the brain interpret those signals that are coming in. It's similar with optogenetics. The brain, it'll be a learning curve because the brain will be seeing new information or receiving new information that it hadn't seen before. So it will have to learn to understand what is being sent and process that to make meaningful sense of what is being delivered and the messages that are being sent. So that could be very interesting. And then assistive technology also, and many of you probably already are using screen readers and navigational apps. And there's a continual pipeline of improvement for smart canes and self-driving cars, and again all these things that are coming down on the horizon that are just really incredible. That there's so much progress going on in technology to assist people with vision loss. So that's a broad overview of where the landscape is. I'm sure there'll be more information presented next about the specific research that CRF has funded. And please, I encourage you, if you have any questions about the information I've shared, to please contact me. Kathi Wagner at curechm.org is my email, or my cell number is 312-550-8122. Happy to talk with you at any point in time about this presentation or anything else CRF related. Our programs, our services, fundraising. Again, we are 100% philanthropically driven, so we do not receive any government funding. We don't charge much, if anything, for our services, except for some of our larger conferences. So really, all efforts, all these research projects that we're pursuing couldn't happen without philanthropic donation support, contributions from people like you. So we're really grateful for everything that you do to help our organization help you. So thank you very much and enjoy the rest of the presentations today. Thank you.